Animal viruses are going to be viruses that infect animal cells. And for these, they don't have to penetrate a cell wall, which would be around the bacterial cells or the plant cells. So it's going to be a little bit easier for them to actually get into the cell, okay, because animal cells don't have the cell wall. But nevertheless, they do still have to get inside. So animal cells would have the specific receptor proteins on the outside that are going to allow those specific viruses to infect them or to attach and get to the inside of them. Once that happens, there's a variety of effects that could happen from that. So some animal viral diseases happen to be what we call acute diseases. And what we mean by this is that these are diseases that we get, or viruses that we get, and we have them for a relatively short period of time, but in any case, our bodies are able to fight them off. So that would be something like the flu virus. So with a flu virus, we have it for a certain length of time, maybe 2 weeks. Our bodies fight it off, and then it is gone. We also have ones where we have what we call a chronic infection. And a chronic infection is something that you're not going to be able to get rid of. This is going to be something that once you contract it, you have this really forever. This would be something like the herpes virus. Now with these, they can cause what we call intermittent symptoms, which means the symptoms may come and go. So we might have a period of time where we don't show any symptoms at all, and then you might have a period of time where you actually do show symptoms. In other cases, you might show symptoms for the entire time of your life. So from the time you get it until you die. It's also possible to have asymptomatic viral infections. And what we mean by that is that individuals that have these types of viral infections, they don't show any symptoms at all. So even though the virus is able to survive and replicate inside of their cells, again, there's no effect. So it's not really detrimental to the individual, but that individual could still pass that virus on to another individual. The last one is that we do have some viruses that have what we call oncogenic effects. And oncogenic effects would mean that they are capable of causing cancers. And so the human papillomavirus is one example of this, and that is one reason why the vaccination for that particular virus has been pushed so much, because it does have the potential to cause certain types of reproductive cancers. If we were to talk about the plant viruses are going to have a little bit of a tougher time getting into cells because they have to go through a cell wall that's around all plant cells. And so in the case of plant viruses, a lot of times in order to enter, they might need some type of cell damage or help to actually get in. So there might need to be some kind of physical damage to the cell wall. So this could be from insect chewing, or this might be from you using clippers and cutting the branches, breaking the branches, something like that to actually enter. Once they get in, there's all different types of effects that they can have. We could have things like you see here, which is tumors. Okay, so we can get tumors in plant organisms. We could have what you see up here, and in this case I'm pointing to the brown or the dead areas. That's what we call necrosis. And so we have cells that actually die because of the virus. And then sometimes we just see kind of a loss of color. This is really a decline is what we're seeing, where the cells start to grow less and less and less. And so the cells are growing less, or the cells are able to do less than what they were able to do before. And so there might be a decline in the photosynthesis, and that then provides less energy, and we start to see the yellowing that you see in this particular example. So there's a variety of plant viruses. Some of them have big economic impacts. They can ruin entire industries. The papaya ring spot virus is one of notable importance that essentially wiped out the papaya industry in Hawaii for a period of time until they came up with some varieties that were resistant to that. So that's a look at just a variety of viruses. There are ways certainly to treat viruses. Some viruses we have really good ways or good tools to treat them, and then others it's a little more difficult, or we haven't quite figured it out yet. But one popular way of treating viruses, and probably the most popular way of treating viruses, is through the use of vaccinations. And if we talk about what a vaccination is a vaccination is basically a treatment to help our cells build immunity. So what we're doing is we're kind of training our cells ahead of time to fight this virus infection or get kind of a jump start on this particular virus infection. And so this can be done with a variety of things. This could be done by treatment with like the capsid proteins from a virus. In some cases is actually treatment with a virus that it's a live virus, but a live virus that is what we'll call disarmed. So in other words, in a lab, they have taken the virus and they've removed kind of the disease or symptom causing portions of it, and then they use that to infect individuals so that they can build immunity to the version that would actually cause the symptoms. So that's a couple of the tools that we use to produce vaccinations. Vaccinations are pretty effective, but in some cases they're not entirely effective. We also have what we call antiviral drugs that are used. And antiviral drugs are drugs that hinder virus replication. And so in some way they help to prevent the virus from actually replicating, which usually this is going to be what it does is it hurts the viral proteins or keeps the host from actually making the viral proteins. So that virus is not going to be able to replicate, transmit to other cells. One example of this is Tamiflu. One thing that is done a lot with virus treatment is the use of what we call cocktails. And the idea between cocktails, it's a mixture. It's a mixture of drugs. And the reason we use a mixture of drugs is because viruses, because they replicate so frequently, they do tend to evolve pretty quickly because they mutate so much. So it's very easy to have them kind of evolve to be immune to a particular drug. So by using a cocktail or collection of drugs, you get more of a widespread, what we're saying here, kind of hindering of virus replication. Just overall works a lot better, and it will take more time for a particular viral strain to probably become or evolve to be immune to all of the variety of drugs that we happen to be using. The last things we want to talk about are the prions and the viroids. And these are even smaller than the viruses themselves. Both of these are also going to be acellular, so they do not contain a cell structure. They don't have a membrane, they don't have organelles, all of those different pieces. And really both of these are just single molecules in themselves. So the prions, these are what we call infectious protein. And what this infectious protein does, and this is what's being shown in this picture right here, is this infectious protein is going to cause misfolding. And so it causes misfolding of the host's proteins. And when it does that, then the host is going to have dramatic effects because those proteins had specific roles that they were playing inside of the host. The most popular example that we have of a prion disease is mad cow disease. As far as the viroids are concerned, these are composed of just a single RNA strand. And so it's single-stranded RNA, but that single-stranded RNA does tend to be catalytic, and so it has enzyme properties, which means that we refer to it as a ribozyme. And these themselves, we don't know to date of any that infect humans, but there are numerous ones that do infect plants. So they're able to replicate inside of plants and cause specific disorders. Okay, so when we're talking about viruses and prions and viroids, again, a lot of controversy as to whether these things are actually living or not. But regardless of that, they are able to replicate inside of a host organism, and then they can be transmitted to another cell. They replicate very quickly, within their host organisms, which means that they are able to evolve rather quickly. And so remember that evolution is based on variation. These do not have real good replication machinery. They're actually utilizing the host's replication machinery. And so there's not a lot of proofreading and things like that take place when they replicate. So that's going to lead to a lot of mutations, which is the basis for natural selection to occur.